Very few scientific discoveries are actually true breakthroughs, but this one qualifies, as scientists at the Scripps Research Institute created what they call alien DNA by expanding the fundamental alphabet of life. Joining me with why they did it and what this discovery could lead to is my guest, Court Turner, president of the San Diego company Syntherex, that now owns this new technology. Court, welcome.
Thank you.
Now, we've got some animation to explain in very simple terms what DNA is and how the scientists changed it. Could you describe what we're looking at here?
Sure. So DNA are the building blocks of life, of the genetic code, and so usually referred to as by their letters A, T, C, G, which you see here. And so what we have done, and what Floyd Romsberg's lab has done at Scripps, is expanded that by adding an X and a Y. And so essentially that is the basis of life, and we've expanded the genetic code by 50%.
So this X and Y that they added, these new building blocks, chemical building blocks, what are they made of?
So they are chemical moieties. So they look like some, if you went to chemistry class, it looks like any others. But they have a very specific structure where you have this compound linked to a sugar, linked to a phosphate. And so it takes the same general structure as ours. And the important part about Floyd's technology is that it looks just like a natural base, and the cell thinks it's a natural base. But we've been able to expand it. And so he's been very, very specific over the last 16 years to make it act that way.
And the other part of that is these extra X and Y DNA, the building blocks, were injected into a bacteria, I understand. And that bacteria actually replicated on its own. Is that correct?
That's correct. So once we got these unnatural DNA into the cell, no other part of the cell was changed. The regular cell machinery, which is called the polymerase, that replicates the DNA in any normal E. coli cell, looked at Floyd's natural or unnatural bases and kept replicating along.
Why create a part natural and part synthetic organism that can replicate on its own?
So it's really about diversity, so it becomes a tool. So in our case of the company Syntherex, it's a tool for developing new drugs. And so if you think of the genetic code as storing information and proteins, which they code for as retrieving that information, it gives us more information. So right now there are 20 natural amino acids. By expanding genetic code with 2 additional bases, we actually could access up to 172 amino acids, making proteins that might become future drugs for healthcare purposes.
So how would that work in the body? Would you replicate the cells in the body, or would that be just the drugs that you're doing?
So we would use the cells as the source of the product. The product would be taken from the cell and modified to be appropriate for human administration, and then by the time it gets to the human, the system itself will have been long gone.
How do you respond to concerns that scientists don't completely understand natural DNA right now, so it might be reckless to create a hybrid forms like this?
Yeah, I think it's all about the safety measures. So the system itself has a built-in safety. So these unnatural base pairs can only be replicated if the scientist in the lab feeds it what it needs, this chemical substrate called an unnatural triphosphate. Once you stop feeding it the unnatural triphosphate, the cell will no longer replicate the unnatural DNA, and it'll revert back to its normal form.
So does that answer the question of could this sort of hybrid DNA escape from the lab and somehow infect people or animals?
Yeah, I think it does. It's literally impossible with this system for that to happen. There is nothing in nature, nothing inside the cell that could ever produce what it needs in order for it to replicate. What
are the next steps here? So you've got this, you've started the new company, there's patents, I understand, on this. Where do you go from here?
So from here, we optimize the system and we start using it to show that it can be used for developing new drugs. And so it's all about diversity. So when you're going after new drugs, whether antibiotics or new cancer drugs or new vaccines, you want to ask as many questions as possible up front and get the best answer on the other side. And we've learned that the cell is the best tool for asking the questions and for giving us the answers. And before, we would ask a cell, go back to the lab, figure something out, go back to the cell, and then back and forth very sequentially. This now, because the system has this extra genetic code, we can go from asking a question to getting an answer in a very short period of time, all in the same cell system.
All right. Well, Court Turner, I hope you'll keep us posted on this. Very exciting. Thank you.
Thank you.